at ICGB. So I would like to start with I'm Dr. Pawan Jutu, working as a group leader at ICGB at Omics of Algae Group. And today I'm going to talk about algal chases, the future of biomanufacturing. Because the previous speakers have introduced microalgae, how to characterize the microalgae, different roles of microalgae, how it is. But it is the need of the hour that you have to understand how the microalgal cell behaves in terms of how it is able to biomanufacture different compounds of interest, how it is able to sequester wastewater to useful products. So everything what is algae doing in nature is need to be understand, and how we are trying to understand using molecular tools, which is known as multi-omics or integrated omics, to understand the cell physiology and the nature of that. Before going ahead with my talk, I would like to have a small brief introduction about ICGB. So ICGB is an intergovernmental international organization which have different programs, and mainly we focus on areas of research, which is like uh, sorry. Um, Mainly we focus on areas of research, which is infectious diseases, non-communicable diseases, and plant biology, which are three major areas of biotechnology. And I work in industrial biotechnology group, where we are interested in biofuels and renewable energy. So the mandate of ICGB is to cover sustainable development goals, and in that context, we are working extensively on zero hunger, promote health and well-being for all, quality education, promote access to affordable and sustainable energy, which I'm going to talk about, microalgae as a model system, promote innovation and promote global partnerships for the goals. So this is overall context of what ICGB works upon, and we have different programs where we run international fellowships, and also recently we have concluded one of the workshop on algal metabolomics, which tells us how you can reveal different metabolites which are involved in production by these microalgal cell factories. So with this brief introduction, I would like to go for a little bit of introduction, because already the previous speakers might have given introduction in this scenario, where most of the things might have been covered, but I may be repeating it. So on to my left, this is the overall schematic of biofuel generations, where we have four generations of biofuels. First generation is especially from edible crops, which is not at all possible because we cannot use, there is always food versus fuel debate, which cannot be used for biofuel generation. The second one is lignocellulosic, which has been bioethanol kind of thing. Already government has a mandate to blend ethanol, and we are trying to achieve 30% blending or 20% blending by 2027. So this is in progress, and much of work has been done on the second generation biofuels. That means especially lignocellulosic waste, and this is the need of our, because we have to convert the lignocellulosic waste biomass into useful products. The third one, what we are going to talk about, is third generation algae, that is any kind of algae which is non-GMO approach, where you can use this algae to produce many number of compounds, starting from biofuels, biochemical food products, and all those things. The fourth generation is non-edible and also a genetically modified microalgae kind of thing, where you can use to sequester CO2 and also convert into useful products. So this is how the raw material selection of the first component, where you can select based on your generation what you are trying to look upon. And there are other types of things which has to be, where you can see that many the technology of transformation, this is most, and this is the cost, this should be the process where you should work on the cost effectiveness. It may be a physical, thermochemical, or biochemical process, where you can translate the biomass into different compounds, and that compounds may be a bioenergy, biofuel, or any of these things. So the main question is, why do you need biofuels, because you know that fossil fuels are limited. Global energy demands keep on increasing. CO2 release from fossil fuels causes climatic change. We have to have liquid petroleum transportation fuels other than wind, solar, and hydrogen. All this put together, but biofuels are carbon neutral. That means the net carbon energy which has been emitted will be always zero or negative. That is more important. That's why we call it as a biofuel in that context. And this is very important to understand because environmental factors are also the major concern. So this I'll not go ahead. This is just a classification of how you can define algae. There are mainly three types of algae, which is cyanobacteria, microalgae, and macroalgae. And the cyanobacteria are known for their the most popular is spirulina, which is a single cell protein. And microalgae, there are many microalgae which are extensively worked upon, especially green microalgae, brown microalgae, red microalgae, which can be for multiple products, either for carbohydrates, lipids, or any high oil products. The other one, which is gaining much and much attraction, is macroalgae, which will be the fourth generation feedstock for any of these things, because the advantage of using macroalgae does not have lignin content, and so it is easy to break down, and it is rich in carbohydrates. So where macroalgae is now popularly known as seaweeds, or it is used for food industry, which is very important. And so macroalgae can also be an kind of algae which can be used for production of these high value products. So why algae? Because algae, as other eminent speakers have covered, it can produce much higher amount of oil productivity per acre. It can be grown in non-arable land, wide variety of water sources. As I've seen that many people were talking about wastewater, dairy water, or domestic water. So you can use any kind of, especially the microalgae, which can grow in marine environments, is of more useful, because we have a high marine environment where people can grow in the coast area of India, where we have a high coastal belt, and these algae can be easily grown in those environments. And these are not susceptible to anything. So it is easy to grow marine microalgae, which are very important. And also it is used in mitigation of greenhouse gas emissions. A good potential for using both. So, friends, it is always seen that it's not only where microalgae gained much attention for production of alternative fuels, that is biofuels like biodiesel and all, but because in the next slide I'll be telling why we should also look for co-products. At any time, microalgae is able to produce many high value co-products, which has to be looked upon, and that's what we call it as. To understand the algal chassis, what kind of production it can do is very important, and understanding that will help us to understand how these products can be produced, and how and to and how you can extract these products in a beneficial way. So microalgae has a repertoire of molecules which can be produced, and these molecules are very important for any of the use. Uh, so, what are the challenges? We have seen that there are multiple challenges among the among for biofuel production, especially large scale nutrient supply, recycling, gas exchange. These are the areas which people have been extensively worked, because we need to end, and this is the call. This is the high. That is why due to this harvesting and large scale production, the what we call it as post cultivation or post processing technologies are very expensive. That's why the cost of biofuel is very high. So in order to understand that, how do you produce a viable or a sustainable biofuels is very much important. So there is a review article which has been published in 2016 by a huge group from NABV report, where they have shown that this is a roadmap where if you can follow this roadmap, it can be possible to produce biofuels which are sustainable. And also you can produce uh, a gallon at a cost of $7.5 per gallon, which is how they cost it. So Basically, these have the discovery part is the one which most of our labs do upon, and we have the algae of our interest where we try to look about the algae and try to see what that algae can produce, characterize those things for the production of other lipids and all. So cultivation may be open pond or for close to photobioreactors, but in this case, because we are talking about not the high value products, we are talking about a biodiesel, so you can grow in open ponds where it is feasible to grow those algae, and the productivity should be the lipid productivity should be at least more than 25% in 25% of dry cell weight, which is required. Um, Thus, there is a what you call as a benchmark where if you are selecting an algae, how much this algae is able to produce a lipid will be sustainable. That is approximately it should have 25 to 30% of lipid, what we call it as neutral lipids, so that this can be feasible. So harvesting, as I said, the harvesting is the most expensive method along with the extraction. And so if you can reduce the harvesting process, then the overall cost can also be reduced. So at this point, they used electrocoagulation or centrifugation to harvest the cells and also look upon those things. I think this has also been covered by many of the presenters who have been previously talking about. The point, what I would like to think, there is a conversion to fuel or a co-product is more important. The co-products can be multiple. They can be used as fish feed. They can be used as any high-value products in pharmaceutical and nutraceutical products. But these should be extracted so that that cost which is produced, which has been generated from the co-product, can be used for the production of biofuel extraction or harvesting, which ultimately brings down the cost. Thus, the overall cost, if you can look upon this roadmap, the overall cost is which can be achievable by using this system or this kind of mechanism, which now as a tradition pathway is a $200 per gallon can come to $8 per gallon as a biocrude. So this is one roadmap which is shows that biofuel is feasible, but we have to have as many as co-products which can bring down the cost of overall biofuel generation. So at our group, so we do at our group, this is what we call it as B3 algae. The concept is we apply different tools to understand the basic biology of the microalgae, how they are behaving it. And onto my left, you can see that there are three inputs which has to be required for the algae to be given. And the three outputs which are coming out is biomass, biofuel, and high value products. That's why we call it as B3 algae. So basically, it's like a computer. Algae is your central processing system. You need the input like sunlight, carbon dioxide, atmosphere or industrial waste gas, and water, which is wastewater, freshwater, or seawater, and with the nutrients which can be supplied. And it produces a lot of products, that is biomass, biofuel, and high value products. The high value products may be antioxidants, vitamins, omega-3 fatty acids, and biofuels is biodiesel kind of thing. So also you can also, some of the speakers were talking about electricity generation directly into photoelectrovoltaics and all those things. So that's the purpose of generating multiple products using these three inputs into three outputs is processed by algae, which is a waste to wealth kind of thing where you can generate. But we have to understand how these mechanisms are happening. And in order to understand, we take the tool of omics, which is an integrated comprehensive approach combining genomics, transcriptomics, proteomics, and metabolomics for advancement of understanding the molecular mechanism of the cell. So onto my right, you can see there are two flow charts. So it's called forward omics and reverse omics. What do you mean by that? Means if you have a genomic information, and from there, if you're trying to understand the transcriptomic and proteomic, and through that, how the metabolites are getting converted, which can be translated as a traditional approach, where it is used for many of the strains which are sequenced. But what happens is most of the time many of the genome sequences, for example, there are thousands of species, lakhs of species, which are not sequenced. And it's a still an expensive affair to understand, and it needs a lot of expertise to get up with the genomic thing. So it is easier when you find out, when you get a new when you find out a new what you call as a strain, you want to you want to characterize those strains, and the best way to characterize is through reverse omics. That means I don't require any genomics information. I can start from metabolomics and look for what kind of metabolites it can look upon, and then connect with the proteomic and transcriptomic to unreveal what the entire cell is going to happen. So that is called multi-omic analysis or integrated omics. And why it is required means it is not a one pathway you need to understand. It is very important to understand the overall performance of a cell when it's subjected to particular condition. For example, when it is subject to heavy metal stress, what is happening to the system? Because there is a change in glycolytic pathway, there is a change in TCA pathway, there is a change in different pathways. It's a comprehensive changes which has happened. It is not a one pathway alone. So we need to understand how the cell behaves in different conditions. It may be a stress condition, it may be a overexpression condition, or it may be a genetically modified condition. How they are going to behave is more and more important. That's how using these kind of approaches will help us, and the basic exploratory research will help us to understand the overall patterns of the cells. That's why we call it as an integrated omics approach, to understand the molecular mechanisms of cells. Once you understand that, anything happens in near future or later can be a benchmark, and that can be used, outline can be used for understanding what is happening in due course of time. So this is the outline of what we do in our lab. So to make understand, this is similar to what I have shown the roadmap of 2016. So this is an integration of 6 to 7 PhDs who have been graduated from my group. And what we do in our group is we try to understand using multi-omics approach and try to produce these products like biomass, biofuels, and high value products, which is having many importance in terms of, so the overall concept is called algal biorefinery or algal biomanufacturing. What you are trying to produce at a particular condition can be translated to an industry is nothing but it's a biorefinery approach, where you can convert these products, and you can also look for that. So to understand that, what we actually do is we mimic the conditions which are very close to the environment. That means we study that. We do a concept called media engineering, where we don't genetically modify the strains, but we try to plug and play with the parameters. Like, for example, carbon and nitrogen ratio, co-cultivation experiment, supplementing with CO2. Somebody was talking about CO2 supplementation to the algal cells so that they can grow faster. So light is essential, nutrients, and some kind of chemical modulators where they can phototrophically grow and produce the product of interest. Other area of work, what we also do is heterotrophy. That means we supplement with the cheapest sources. For example, there is a algae which can produce high amounts of omega-3 fatty acids. That's tostochondrites. So in that algae, we supplement, because the glucose is not available, because glucose will be taken off for the production of bioethanol. So glucose will not be available as a feedstock for us. So what we have done is we have used similar sugars like crude glycerol, which is a byproduct of fermentation process, or the fermentation process, where this crude glycerol is a cheaper source, and it's a carbon source by the algae to grow upon. So we use crude glycerol as a source of carbon and urea as a nitrogen, and grown in dark to produce these fatty acids. So this is just like similar to yeast kind of system, where without sunlight we can grow these algae using fermentation, and they're able to produce many products. So for example, it is DHA production. The other one is combination of both mixotrophy and which is called a mixotrophy, where you combine phototrophy and heterotrophy to obtain the product of our interest. So iomics, integrated omics. So once we select the strain, of our interest, we set it parameters, how we are going to subject it, and those case studies I'm going to discuss in due course of time as three different modules, which is B3, which is called biomass, biofuels, and biorenewables. How do we look for different terms? So we will be, the end product is either to enhance the biomass or enhance the biofuel production or the high value renewables. So all these processes, what we are doing as a media engineering approach, will be taken ahead for enhancing these products. And there are a number of products we are working in a lab. For example, astaxanthin, lutein, zeaxanthin, ficoxanthin, EPA, DHA, tocopherols, the alternative substitute for vitamins, and also other biofuels. So to understand this, we also do a basic research of understanding all these systems through integrated omics, so that these systems can be understood, and when there are any modifications that can be translated or looked upon, why there are changes or why there is a loss in the product. So this helps us to understand the background, and iomics gives us a clear picture or outline how the cell, these molecular microalgal cell factories are working for the production of these products of our interest. This is the overall view of what we are, and I am displaying the number of papers. All the papers are published. So this is the published data and it's available. If anybody is interested, I can definitely forward. So first, so as I said, like we'll be talking about biomass. So what is the context is here, because we know that when microalgae is subjected to nitrogen starvation, you can produce larger amounts of lipids, neutral lipids. But the challenge is we are going to compromise on our biomass. So is there any way that your biomass will not be compromised, but your lipids also can be produced, or high value products can be produced? So we developed a concept of using carbon nitrogen ratio in order to understand and supplement carbon to these microalgal systems, so that they can we can evaluate the carbon concentrating mechanisms that will contribute for the regulatory networks of this for increased cell biomass. So I will give you two examples. For example, many people were talking about diatoms. Diatoms cannot survive at high CO2 concentrations. Why? Because they are present in the oceans, and when we subject to the diatoms to high CO2 concentrations, they try to not to grow, okay, or die. So basically, because they are very strong with carbon concentrating mechanisms, which is also known as CCM mechanism in microalgae, where they try to evolve a system where even a small amount of carbon dioxide present in the water, that is 0.02 to 0.03%, that is 300 or 200 ppm, can be absorbed through these carbon concentrating mechanisms, and that can be converted into useful products. Thus, these diatoms have developed a system, because in oceanic waters, we will not have that much of carbon dioxide continuously in flux. So in order to understand how these behave, and we want to understand and compare the difference between how, when we supplement with higher carbon, what is happening when we are supplement. So we want to know what exactly is happening within the cell. In order to know that, we have used a unique system of uh, uh, chlorella species, where we try to understand how this chlorella species is behaving in terms of how the cells are proliferating. And also we worked on Micromonas gladiatana and other species, where we supplemented with 3% carbon dioxide, which is tend to be high 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 percentage of carbon dioxide. So in this experiment on your right, you can see DLC, which is normal air, 0.03%. So when you supplement with the air cylinder, you will get only 0.03%, that is 300 ppm. When you supplement with 3%, that is around 30,000 ppm, that is 3%. So you can see that there is a drastic change in the growth profile of the microalgae. That means you are getting good number of biomass productivity, good protein, lipids, and carbohydrates, along with the increase in pigments. So what we have done is, because this microalgae is not being characterized, what we thought is we should go ahead and look for different look for reverse omics approach to, in order to understand what exactly this mechanism is doing. And onto my right, you can see we have done a qualitative metabolomic approach where you can see number of metabolites which has been represented on different. And all these experiments will be done in time course. What is a time course? It is important that the relative expression of any metabolite or a protein or a transcript should be done with respect to time. So that's very important. That's why most of our experiments are done at time course. And we know that at a particular time, your metabolite of interest get increased. So these are the metabolites what we have identified in this condition where we have supplemented with carbon dioxide and without carbon dioxide. Where we can see, so they are the onto my extreme right, you can see the fold change. So you can see the number of metabolites which have been upregulated on the top and downregulated on the bottom. So thus we try to evaluate what metabolic changes happens, and we try to illustrate and reconstruct the whole thing using keg mapper and our understanding on the other pathways through a multi-omics approach. And we come with a outline during the normal low carbon concentration or VLC condition, very low carbon. So basically the carbon is fixing through CCM, and it is able to produce the starch metabolism, and other products are being produced, and is also able to produce trellose, which is a storage sugar, in which it is able to enhance the biomass and other products. So what happens? What is when we are supplementing with high carbon, the carbon flux towards lipid biosynthesis and other terpenoid forms like alpha tocopherol, because alpha tocopherols are also important in terms of scavenging the things, antioxidants, and other high value products. So we have seen that at high concentration, the carbon flux is diverting towards high value products, not only the lipids and other things. Whereas in low, it is just involved with growth and other important relevant metabolic pathways. So in order to understand the deeper into that, we have done carbon partition using proteomics and metabolomics approach. And we try to understand, this is the overall schematic representation, how this is done in VLC and HC. We also maintain a dark period of 8 hours and 16 hours of light period, which is shown in gray color dark period and white in is light period. And the the green colored pathways are the one which has been upregulated during VLC condition, and the blue color are the one which are being upregulated with the normal HC condition. So in order to understand that, we can see that most of the when carbon supplementation is high, the growth is also high, and that's why there are many upregulated proteins which are related to TCA pathway and also fatty acid synthesis pathway, what we have confirmed with our metabolomic study. So we have also seen that carbon is getting fixed through Rubisco, and TCA cycle is being active, and that is leading to the carbon flux is getting diverted through pyruvate to all the metabolites of interest, also by isoprenoids and all those things. Whereas in VLC condition, what is happening due to photorespiration, there is the process of photorespiration is hindering the growth. That's why when the air, in the presence of air, the growth of the microalgae is slower. So the process of photorespiration is happening, and the carbon, because of low carbon, the cell is not able to divide faster rate, but it divides. So thus we could find out, identify crucial proteins involved in normal air condition and also high carbon conditions. Thus, we were trying to evaluate. So if you can look for a condition where the photorespiration can be minimized, then it will automatically enhance your biomass. So it is very important to understand that you have to supplement the carbon and nitrogen ratios in a higher amount so that the algae can grow in a faster way, and where there is an additional carbon which is present, that carbon flux is diverted towards high value products. So thus there are different products which can be produced, and these high value products are very relevant for biomanufacturing process. This whole process is called biofoundry because we are trying to understand employing omics approach, synthetic biology approach to understand what kind of mechanism is happening within the cell so that they are able to produce the products of our interest. In this overall process, we are trying to answer why the biomass is getting lower in very low carbon and how when carbon supplementation is provided, how the metabolic regulation changes in terms of metabolomics and proteomics have been revealed, and thus how the biomass is getting increased has been demonstrated in due course of time. So now coming to biofuels, this is a very well-known theory where when microalgae is subject to nitrogen deplete condition, the lipid biosynthesis goes up and photosynthesis and beta oxidation comes down. That is the main reason why your overall cell biomass reduced. So we have done different starvation methods in an indigenous marine microalgae, which is known as Parachlorella kessleri, and this is able to produce more than 30 to 60% of dry cell weight of lipid when it is subjected to nitrogen starvation or any kind of starvation. So but we need to look upon. So how did we answer that? So we have done the same thing because this is a new strain. So we try to first understand the fatty acid biosynthesis pathway, why it is able to produce this much of fatty acids. And we have seen that we have done qualitative metabolomics, and we have seen that the number of metabolites get upregulated and downregulated, and there is a huge flux towards acetyl CoA and acetate, which is activated by acetate, where it is leading to fatty acid biosynthesis. This is why this paper has been published, and it is well documented that there are a number of metabolites which are getting upregulated when nitrogen starvation will, and there is a recycling of lipids which are being translated into neutral lipids. So in order to understand further, we have done, so this is a reverse omics approach, what we have done. So in order to do that, we have done a proteomics, and we have found that they are significantly around 150 to 200 proteins which are being upregulated and downregulated in the stress condition, and these were identified. And also we have done the transcriptome, and on to my right you can see that this is the pathway, the red, which is the upregulated during nitrogen starvation, which is leading to the tag biosynthesis, and we have identified the proteins and transcripts which are getting upregulated. And interesting part is when there is a stress phenomena, the whole TCA pathway does not activate, and there is a semi or half TCA which undergoes, where from succinate to malate, malate to pyruvate, it recycles and it does not undergo all this, so that it cannot waste ATP or NADP. So it is directly converted to pyruvate, and that pyruvate is stored through the KEGG pathway, which is in the plastids through to the tag biosynthesis using Kennedy pathway. So this is how the overall biosynthesis happens when it's subjected, and it's integration of transcriptome and proteome. And we have found certain leads in our transcriptome, and we have validated those leads. And you can see these are the red ones which are highly upregulated, and these are the blue ones which are downregulated. That means when they are subjected to stress condition, few of the genes are downregulated. For example, photosynthesis, as I said, like growth related, or all downregulated, and we have tested that the transcript goes down. That means the overall the metabolic pathway goes down. And these are the pathways which are affect exactly the Kennedy pathway, which is getting upregulated, and other pathways which are related to FAN. So, and this is shown, and this is how we outlaid the proteomics and transcriptomics together, sorry, genomics and transcriptomics together, in order to understand and show the expression of key regulatory pathways. And this is all done at ICGB. Uh, and this is the whole pathway, and we sequenced using hybrid genome technology, and we have done the functional annotation of these genes. And you can see that pink is the lipid biosynthetic pathway, and we know all these pathways are being highlighted, or the identified pathways which are present in Parachlorella kessleri. Now we know the skeleton of these, what are the pathways which are present. Now what we have done is we have integrated all these data. So you can see the yellow spots are the one which are the metabolites, which were upregulated and downregulated. The smaller the spot, that means the metabolite fold change is lesser. The bigger the spot, the metabolites are higher. This is the biosynthetic pathway. The blue line shows that they're downregulated or integrated transcripts, which are both proteins and transcripts, and these are the TAT pathway biosynthesis genes which are available. So these integrated pathways show you conclusive pathways, what is happening in the stress phenomena when it is subjected, and how your neutral lipids are being produced, and it will give you a complete picture how your cell is behaving. Thus, we use approach of integrated omics to understand what kind of pathways are getting upregulated and downregulated. Among the previous one, what I showed, these are the pathways which are integrated, and this is real-time data where we have used the experimental data to reconstruct the pathways, and now we understand how this behaves. So when tomorrow I'm changing any condition, I can, from these pathways, I can know what is happening at the molecular level, so that I can fine tune my pathways, or I can correct my initial things, so that I can produce my product of interest. Uh, Final thing is biorenewables. So biorenewables is very important part. So on to my left, I was talking about heterotrophic growth. So we supplemented with crude glycerol, and we have seen that there is a high production of DHA and also squalene in this microalgae. And the same thing has been done. Integrated omics have been done to understand, and we have demonstrated that number of metabolites, number of pathway has been regulated, so that we can produce a DHA of our interest. This is very interesting because many of you were talking about diatoms, and this is a new approach where we employ diatoms and see what we can produce, and we are able to produce large number of, especially the fucoxanthin and omega-3 fatty acids, the combination of which is very, very important, and it is used for the treatment of anti-cancer and stuff. So we have done the same thing. We have done the screening, and we have developed a media engineering approach, how to grow at a large scale this thing strategy, and how you can replace with the cheaper sources which can be sustainable and cost effective. Thus, we used omics to understand the overall mechanism, and also we have shown that the natural extracts of these omega-3 and fucoxanthin were tried on some of the cancer lines, and they were seen to be very, very effective in terms of, so they are potential green medicines of the future, what we can look upon. The last one is tocopherols, which is very important. Again, we try to understand employing omics, and this is a native strain where we supplemented with CO2, a new approach where we played with carbon and nitrogen ratio in order to produce tocopherols. And this is the one of the few strains which produces tocopherols, and tocopherols has vitamin E, which is very important. And vitamin sources are produced from the plants, but it can produce 10 times or 15 times more than the plant sources, which is very, very important. And this is a product which is also required. So we have developed a technology where we can use a media engineering of supplementing CO2 and limited nitrate to convert into high value products like tocopherol with other value added products. So my final slide is that we are able to also generate fourth generation. So when you fourth generation biofuels are nothing but if you are able to highly engineer, because we know that next 10 to 15 years we have to get a lot of biodiversity guidelines to in order to do GMO based algal sequestration. But these technologies can be developed because highly engineered product specific photosynthetic organisms in non-freshwater solutions can be found out, and this is where how you can develop solar converting systems on non-agricultural lands and convert into high value products of the interest. This is a kind of a future perspective. And thank you very much. And with this, I'm going to thank all my collaborators, national and international, and all my students and fellows who are involved in making this um, uh, research fruitful. Thank you.
Thank you, Dr. Pavan, for sharing your work with us. This has been a really insightful session, and your work in this domain would have surely motivated our attendees. It's great to have a better understanding of algae chases and cell mechanism, also the use of an integrated approach for enhancing the production of P3. So we can now move on to address the questions from our attendees. So the first question we have is, um, considering algae biomass as the feedstock, which among bioethanol or biodiesel is commercially feasible?
Okay. So that's as I said, like bioethanol, you need carbohydrates as a source of algae. So seaweeds will be the best for bioethanol, and also we can also